Why do we need to know all about the equipment before we start our cell culture study? By knowing the equipment and their names, it will help the beginners to familiarize with the description of the methodology when reading the original articles related to the studies and theses. Not only that, by knowing the equipment, this will help the users to use the equipment correctly and maintain its good condition. Hello students, in this session, you will learn about the name of the equipment that are commonly used and available in cell culture lab and also the function of the equipments. Let's learn. This is the picture of pipettes. These are the pipettes that normally used in cell culture study. There are many types of pipettes and manufactured from different companies. The pipettes are defined based on their volumes of the liquid that it can suspend. So we have different volumes such as 1 microliter, 10 microliter, 20 microliter, 100 microliter, 200 microliter, 1000 microliter and as well as 5000 microliter. At the end of the pipette, there is a narrow end and it is where you connect to the tips for suspending the volume. Before using the pipettes, you have to make sure that the equipment is maintained and calibrated. The arrow in this picture shows the pipette gun. Pipette gun is normally connected to the sterile serological pipette to withdraw certain volume of fluid. The multi-channel pipettes are used to draw the desired volume into more than one channel in a plate. It is commonly used to transfer media, drugs or liquids to 96 well plate. The 3 ml pipettes usually comes in a sterile package, so you can use it to withdraw drugs with a volume between 0.5 ml up to 3 ml of liquids or drugs or PBS or any other liquids. Biosafety cabinet is the place where we do our cell culture work under aseptic technique. The UV light is built in to kill any microorganisms that is present on the surface. Before using it, you need to clean the internal area or the surface area in the BSC with 70% alcohol. Do not leave the BSC on UV mode more than the required time. Make sure you know the buttons and how to switch on the BSC before using it. You can ask the staff or the lab technician for help. While doing your work, any fluid spills such as media must be wiped immediately with 70% alcohol to prevent contamination. Make sure you clean the working area in the BSC after use and turn on the UV mode to ensure the BSC is clean for the next user. This is a picture of a liquid nitrogen freezer or cryostorage. For longer preservation of cells, we need to keep the cells at lower temperature. In our lab, we keep the cells at minus 186 degrees Celsius. Storing cells at this temperature will prolong the viability of the cells. Usually, for cryopreservation, liquid nitrogen is used to bring down the temperature. There is a detector to tell us if the temperature goes up. So you must be alert about this. Maybe the liquid nitrogen might need to be filled out or something might go wrong with the equipment. So do not ignore the alarms or the beep sound. In cell culture studies, temperature plays an important role in storage or cryo-freezing cells for long-term use. On the left-hand side is minus 20 degrees Celsius freezer and on the right hand side is the minus 80 degrees Celsius. In cryopreservation step, you need to reduce the temperature slowly by keeping at minus 80 degrees Celsius overnight before storing the cells at minus 186 degrees Celsius for longer storage. So this is the picture of incubator. Incubator mimics the human body for cell culture study. So there are many types of incubators. Most human cells require 37 degrees with 5% carbon dioxide supply for growth and maintenance. You can set the temperature according to the requirement of your cells. Some cells may need lower temperature. Example will be 27 degrees, but some cells require, or most of the cells require 37 degrees for growth. Besides that, you need to know whether your cells require carbon dioxide or not. Some cells do not require 5% carbon dioxide for their growth. Before using the incubator, ensure the incubators are cleaned up first and fill up the tray at the bottom with sterile water and make sure the temperature is stable.